Hello, in the second exercise of this lab, we are going to use again blast, but in this case we're going to use DNA, not protein. And here you will have a section of DNA and you have to use it to find a sequence. So again, I'm just going to walk you through very fast because it's very similar, but now you just have to go to the nucleotide section, not to the protein section, copy and paste here your section, the DNA that you want to get in. You have to select this one. Later on, you click here in blast, and then remember this can take you from 10 seconds to a minute while the page loads, depending on how busy is the server. And if it doesn't play right away, you can, after 1 minute, you can refresh it, and in that way, it will give you the information. So you will get again a description. Here it will give you a graphic summary, the alignment, and the taxonomy. So the different groups that have that type of DNA sequence. This I'm just going to go very quickly because you have seen this graphic representation. Now this is for the DNA. Here you can find the alignment again. But remember this again, this is for DNA, no for protein, and you can going through all the different links, hyperlinks to know more about what you are seeing. For example, here you can see the small section of DNA that I sent you, how similar it is to the other one. Again, here you also get an e-value that is very important. And here are the names of the genes that you picked. Here you have the accession link. So here you can see it in red. And just for you to know, similar to the previous assignment, maybe in a test I can ask you what is the expected value, what is the score, or all this information. So make sure that you know how to do. Here you can focus on trying to get the gene. So here you can have again the accession, the locus of the gene, where it's located. Here they tell you that this is from a human. Here you can get again the FASTA that they already explained it before. So here you have the entire sequence. Remember that DNA sequence, the protein will be much smaller in terms of sequence, while the DNA will be at least 3 times longer. So sometimes to have this FASTA can be very useful. Now the second part of the lab, I'm going to show you how to compare different DNA sequence, and you have to go to this link. Later, you have to create a sequence. Then you copy that sequence and you give it a name. So you can give this name here if you want. If you make names that are very long, the software is not going to accept it. So just make it not more than 20 letters. They will tell you when they are not too long. Then copy the sequence. So this sequence you can copy and paste it here, and then press OK, and the information will appear in a new window. So here you can see the first sequence that I add, and here you can see another sequence that I add. You will have to compare these 4 sequence, 1, 2, 3, and 4. And just an advice, these names are too long, so you can just call these names, for example, Homo sapiens, has maybe these letters here. This one is too long, so you can call it Homo sapiens PTC no taster. Here you can maybe, if you want, just something, a part of the name that make clear what you are comparing. But remember, not the entire name. And here you can find the different sequence. So this is the thing that you have to copy and paste, and I color coded for you. And here in green, I upload twice the Homo sapien paste. I can delete it, or I can just unclick it. If I don't click this one, yeah, I will only compare the ones that are in blue. And here, as I said before, you decide what are the things that you're going to compare. You can compare the phylogenetic tree or the sequence base-to-base sequence. An example of what we have when we compare the phylogenetic tree, and here you can pick the cladogram. Here, this will be, it will not be sequence 1, 2, 3, and 4. It will be the names that you give, and you have to upload this picture as a screenshot of this and upload it into on Moodle. If you change it to phenogram, this will look like that. Again, these names will change, and they will be the names that you had. Again, take a screenshot and upload it on Moodle. And here, this is the other type of comparison that you also have to upload into Moodle. The one that is the clustral analysis. And here, if you see, there are two colors. There is white and yellow. And look how they compare this white. What happened with the white? They're completely similar. And look what happened with the yellow. They can vary. So the yellow highlight denotes mismatch between sequence. And here, for example, in this case, that are all yellow, we only have one because in this case, the Homo sapiens have a piece of a sequence that the other ones don't have. So this is why the other ones are in. Please, if you have any doubt, just let me know and I can answer any question. Have a good day.